In this video, we will talk about sampling. Sampling is selecting a subset of a population. Why would we sample someone from the population? Well, if we want to know something about the population, and usually we cannot measure everyone, then we can select a subset of that population to study. If we want to know something about all humans, we can't measure all humans. We select a sample and study them. Or if we want to know something about all dogs, or about all rats, or about all cells in an organism, or all cells in one tissue, we select some of them. This is our sample. So sampling is the selection of a subset. And sample is the subset of the population that you have selected. Now, which part you select, how you actually choose a subset is very important. And that has to do with what is called the sampling method. Before we go on to talk about that, let us clarify some important words. The word parameter, which you've probably encountered already, refers to a population characteristic, which can be a value. One frequent example is the population mean. So the mean value of what you're interested in measuring, the mean over the whole population, is the population mean and is a population parameter. Other kinds of values or characteristics are also population parameters. A statistic is something that you can compute from your sample. It's a sample characteristic, usually a value. So if you're interested in the mean of a measure, of a specific measure, you can calculate that from your sample, and then you obtain the sample mean, which is your statistic. The word estimate refers to our best guess for something. And in this context, estimate is how statistics are related to parameters. So the idea here is, if you're interested in the value of some measure for the whole population, say you're interested in the mean, so that's the population parameter that you want to know, then you sample and you calculate the mean for your sample, that's your statistic. And your sample mean, your statistic, is your estimate of the population parameter. And if you're doing things properly, it might even be a good estimate. So if you look at these little symbols here, you see there's this hat and a hat, and hat means estimate. So mu hat is not the population mean, is our estimate of the population mean. And sigma hat is not the population standard deviation, is our estimate of that. And what we usually do to estimate these is to calculate the sample mean and the sample standard deviation respectively. So this is the sample standard deviation, which is our estimate of the population standard deviation. And this is the sample mean. Remember, this bar here means the mean. So it's the mean of x's. X's are measurements. So this is our sample mean, which is our estimate of the population mean. Our estimate doesn't have to be a single number. Of course, the population mean is a single number, and we can provide a point estimate by referring to a single value, the sample mean. But we could also report a range of values. Instead of saying, this is how much I estimate the population mean to be, you can say, I estimate the population mean to be between this number and that number. So then you're providing a range, rather an interval, and that's an interval estimate. These two ways are both very important and very useful. Now let's go back to sampling. What's the point of sampling anyway? The point of sampling is to be able to use your statistic as an estimate for the parameter. In other words, the point of sampling is to be able to generalize to the population. Use your specific sample and whatever you can calculate from that in order to draw inferences about the population. So generalization is what you're usually after. This generalization is not necessarily valid. If you pick a subset of the population, that is, an extreme subset in some sense, then your estimate based on that sample will not be a good estimate for the population parameter. The generalization is only valid when it is based on a representative sample. What is a representative sample? It is one which accurately reflects the characteristics of the population with respect to whatever you're interested in. So if you're interested in finding out what the average height of human beings is, if you sample only Norwegians and Dutch, you probably aren't going to get a very good estimate. One obvious question to ask now is, how can we ensure that our sample is representative so that we get a good estimate that we can generalize to the population? Unfortunately, the answer is it is not possible to ensure a representative sample, but there are many things we can do to maximize our chances of getting a good representative sample. Let us now turn to the different ways of sampling in order to somehow understand and better answer this question. At one end of the sampling continuum lies what people call a convenience sample. It is literally called a convenience sample, and it is what the word says. It means you just use whoever happens to be around, whoever is available or accessible at the time you need to conduct your study. Because there is no guarantee that whoever happens to be available is representative of the population, this is generally a biased sample. Biased means we do not expect the statistic of the sample to be good estimates of the population parameter. At the other end of the sampling method continuum is what we call a random sample. The definition of a random sample, and the only way in which a sample can be said to be truly random, is when every member of the population has exactly the same probability of being sampled. This means one must have access to the entire population and sample with a method that gives equal chance to everyone. In this way, we can obtain an unbiased sample. And the reason is that the only thing that affects representativeness is chance. It's still possible to get an extreme and unrepresentative sample by sampling randomly. That would be the result of bad luck and cannot be avoided. One other word that you may encounter in connection to sampling is the word stratified. So you may hear about stratified samples. A stratified sample is not a third option. It's not something that's different from convenience or random sampling. A stratified sample is when you first split your population into parts that are relevant for your measure in some way, and then sample from each of the parts. These parts, these subpopulations, are called strata, and the reason this is done is because in some instances, sampling can be made more efficient. So you may be able to achieve a similar level of representativeness with a somewhat smaller sample. For example, you may frequently see stratified sample according to sex. So sampling boys and girls or men and women separately, or sampling separately from urban areas and rural areas, or sampling separately from different countries or different species or different other kinds of things. In this way, you can ensure membership of all strata into the sample without having to resort to very large sample sizes. You can have stratified random samples and stratified convenience samples. That depends on how you choose your individuals after you've decided to partition your population or not partitioning. So a stratified sample can be unbiased or biased depending on the sampling method within the strata. When is it possible to generalize from our sample to the population? Well, it's possible only when our sample is representative, but we don't know when that is the case. To know that we have a representative sample means to know that our sample matches the population. In order to find out if that's true, we have to know the whole population, but we don't. If we knew the value of the parameter, we wouldn't need to sample in the first place. So we can never know if we have a representative sample. But we can try to maximize our representativeness, and we do that in two ways. The first one is to have as large a sample size as possible, as practically feasible. The second one is to use an unbiased sampling method, that is random sampling. How large is large enough? This is a very difficult question that doesn't have a specific answer, but is exactly the kind of question that statistics can help answer. A larger sample size is always better than a smaller sample size. But there comes a point that further increases in sample size have very small relative benefit. The actual sample size that you should use depends on how precise you want your answer to be. How far off from the population parameter can you tolerate your estimate to be? How wrong do you want to be? You can calculate the confidence that your sample can give you depending on the population variability and your sample size, and we will soon see how that's done. And then we have to find out how to sample really randomly. The only way to sample randomly is with a procedure such as drawing lots or using random numbers. The Lotto procedure with all these little balls in the big rotating bolt is one method for random sampling. Computer-generated random numbers are also fine. However, this is very easy to say and very hard to actually implement, because if you want a random sample of every human being or of every Norwegian citizen or of every child with some specific sort of disability, it means that you actually know them all, have access to them all, and can randomly pick some of them, and those that you pick will be willing to contribute to your study. This is very unlikely to happen. Random sampling is possible and customarily done in controlled situations. For example, in intervention studies and medical studies, we randomly assign participants to conditions. So we choose completely randomly by drawing random numbers which of our participants will receive intervention or medication and which won't. This maximizes the generalizability of our findings in this controlled experimental situation. In observational and naturalistic studies, it is very difficult to achieve actual random sampling, and we try our best to minimize the bias in our sampling methods.